prefer this. Okay. So I would like to introduce myself. My name is Akif Eskalen. I am Professor of Cooperative Extension Specialist at UC Davis, Department of Plant Pathology. A little bit background information about me. I have been working and involved with grapevine diseases, not only grapevine, other fruit crop diseases since 2001. When I first came to UC Davis with working with Dr. Gubler. Some of you may have heard about him. And I worked with him about 5 years, management of the grapevine diseases, and after that I took a position at UC Riverside to work on avocado and citrus diseases for 10 years. And when Dr. Gubler retired, I took over his position at UC Davis, and then since 2018, I have been working on these trials. So today I'm going to give you a little bit more information about managing common grapevine diseases, including the powdery mildew, botrytis, and if I have time, I would like to talk to you about grapevine trunk disease as well. But we will see how much time we are going to have. Please let me know last 5 minutes how far I am going, so we can go from there.
Dr. Eskalen, you have as much time as you want.
Oh, that would be dangerous.
No limit on time.
I gotta tell you. Okay, well, the most important part is how am I gonna entertain these beautiful people? Okay, so is it working? It should work, huh?
It should be. It was. Oh, it's not the time.
So,
Well, we can advance for you.
of course. So what I'm going to start with is that the introduction about my laboratories. You will get better understanding what we do, how we do. And after that, I'm going to get into the powdery mildew, botrytis, bunch rot, and trunk disease. And I'm going to leave you with the conclusion of what we said. Next one. So as extension specialist at UC Davis, this is the system that has been established more than 100 years ago as UCANR. University of California is a land-grant university. When it was first established more than 100 years ago, it was promised that this university needs to or has to work with the applied research, address the problems, issues with the growers that have been facing, and then try to solve that problem. Since then, we have been working on any of the crop management, diseases, establishment, whatever you can name it, the university has been working it. I'm proud to be part of this cooperative extension system at UC Davis. The way that we work is that my lab has a diagnostic laboratory to identify if there is any new pests and diseases. But we focus on the now fungal disease of any crop that we have been working on. The way that we work in this system is that so when we receive the symptomatic plant parts, so what we do is that we make isolation on special media, as you can see in that area, the culture media. In that culture media, we culture them and then have them grow in the media. When we know what is look like by their morphology, we identify them by their morphology. But nowadays, we have advantage of using the DNA extraction, looking for their DNA fingerprint, and then we also do the DNA extraction, species-specific identification using all kinds of fancy identification methods, and then at the end, we are able to sequence their genome and then identify them based on their either part of the genome or entire genome of the diseases that we have been working on. So this is what we do. So this identification or diagnostic service that we provide to identify if there's any new diseases out there that needs to be identified and also studied or develop a research. That's how we do in that system. Let's go to the next one. So I'm going to start with the powdery mildew. Powdery mildew used to be known as Uncinilla necator. initiator. And then now it has a new name, Arisaiphe indicator, which is the new name that was established, and then that's what we know. This disease is native to North America. It was first identified in the East Coast. Later on, it just moved to the West Coast, and then after that, it moved to the Europe. So this is one of the diseases that actually Europe got it from United States. And why does it matter? It is matter. This disease has been established here for a long time. That means it is adapted. This climate and this the part of the native range of the grapevine in United States. Because of that, it is little bit different managing this disease than the other diseases, because it has already established here for a long time. It produced a special structure which is known as a cosmothecia. This cosmothecia is first start with the brown color, now turn into the black color when it gets mature. So this is the individual structure, which is the sexual structure of the pathogen. And then this is the very unique part of it. And then this is the kind of like the sac that has 8 ascospores, which is the kind of the seed of the pathogen. When the springtime comes, those fruiting bodies release their spores in the air. Then your shoots start bud break, and then from now on, and then move into your plants. The next one. When it first, there is no fruit at the beginning of the season. When it first established on the leaves, when first it came into the leaves, it's very difficult to identify. It can show up like the yellowish color, as you can see right here. It's a very early stage. It's very important to identify in early stage that you can start your management program. Next one. Later on, it turned into this stage. When it turns into this stage, by the time you start seeing that one, it's kind of a little bit too late for putting the preventative management system. But I'm going to go through and then tell you what kind of management system that you are supposed to get in. Next one. Later on, if you cannot control it, it starts with the powdery, which the name comes from, like that. Next one. And then turn into this one. Okay, entire plant. So, because the pathogen cover on the surface of the fruit development stops in certain time. You cannot get the fruit that will be useful. Wine grape growers cannot handle more than 5 to 10% the severity of this pathogen, because nobody's going to get it. So this is the life cycle of the pathogen. When single conidia, which is let's say about the spore of the pathogen, lands on the leaves, within a short time it just germinates like a seed. And then it produced a germination tube. And then after that, it produced a structure that we call it aprosorium. Later on, that aprosorium turned into the haustorium, which is a structure that can penetrate into the tissue. The way that it penetrates, it just makes the swollen, bigger tissue that can forcefully penetrate into the tissue. It doesn't require any wound. It can forcefully penetrate. That's the dangerous part of it, because wherever it lands, it can colonize into the plant. And then as soon as it colonizes, it starts taking the nutrition from where it's penetrated, and then it starts producing from one single conidia to the hundreds of conidia. How long? That's the problem. Within the 1 week approximate the time when the temperature and humidity is in the right time, which this year we have been having a most suitable time for powdery mildew to grow and colonize. If you have working with the wine grapes or table grapes, doesn't matter. you have been probably seeing the high rate of the powdery mildew because of the climate has been perfect, ideal for this crop. Another problem is that when you apply whatever the preventative application that you are applying, if you cannot manage from the beginning, within the 1 week, you are going to have 1 to 100 again. So that requires you to be constantly be on top of it, management practices that you are going to go. My lab has been running not only in vitro laboratory, but also in field fungicide screening trials for a number of the years. So these trials has been run by Dr. Gubler, my predecessor. Now I took over that the experiment. So what we do in this trial is that when we have all the either individual or combination of the fungicide, biocontrol agents, or organics, put them in a field trial in a scientific manner, and then get the results. That results could be utilized by companies who is looking for registration or results from whatever it is, or pest control advisors getting an idea what kind of chemical has been working, what can I apply, or growers to get an idea about that. That's the whole purpose of doing these trials. We set up this experiment. You don't need to take a note. I will show you where you can find this information. And then set up in a randomized block design on the vineyard, and then we set up the experiment. During the growing season, we also monitor the climate or weather data, which is very, very important for disease development in any disease that we have been working on it. For example, this is the 2024 results. So this is showing what does the temperature look like during the growing season from April to the end of July. And then these are showing the precipitation or rain event that we have it. Why is it important? It is important because disease is also being affected by the climate or weather as much as we do. Actually, they are being affected more than any other living organisms. It's either favorable or unfavorable. So during my talk, I'm going to a little bit get into the more information about what those are. We also monitor the risk index, which is the index that was developed by Dr. Gubler for a long time ago, which tells us that under what weather conditions that you have to worry about disease pressure. If the weather conditions are so good, disease pressure is going to be so high, you don't have to see the disease because disease is everywhere. But climate or suitable conditions are the one is going to tell you when it's going to be high or low. I don't know if you are seeing those risk index results moving around. There's a website in UCIPM that you can get the information that will tell you, hey, this week the disease pressure of the powdery mildew is going to be 100% or 80%. That tells you and gives you an idea what kind of the plan or program that you are going to use. Are you going to use the soft chemical that works slow and sustainable way, or you have to use the hard chemical if you are not organic to knock it down to manage that part of, or combination of them. So we are going to go into get that one down. So this is basically telling us that during the growing season, these green bars under that blue line, which is the temperature related, that means you don't need to be panic. The low percentage of the risk that you may have in the powdery mildew. So you can continue doing soft chemical from now on. When the risk index get into the red line, that means, hey, the conditions are getting better for the disease. Be careful. Increase your intervals. If you have been applying whatever you have been applying once every 2 weeks, increase it, apply once a week. You can do that kind of management. When it gets above that, that means that 100%, it doesn't matter what it is, it's going to grow very fast. That means that you are going to manage either program, increased management strategies, and you got to be careful about that. This is kind of like giving us an idea that when and how to treat those applications, which is very useful information. Here's the problem of this system. This system has to be established in each microclimate, because California has a very diverse microclimate. East Coast versus Central Valley versus other areas has a different climate, different risk index. So because of that, wherever you are, try to find out where your local index are. That will give you a better idea, so you can go that. If you can afford, you can also have your own as well. In 2024, we have treated more than 50 different treatments comparing with the control. So we run this experiment on campus vineyard, where we can manage the climatic data or climatic area that where the vineyards are, to increase the pressure of the disease, because our goal is try all these chemicals under the high disease pressure. When these chemicals get out with the lower disease pressure, they can work on that one. So that's the whole goal of it. We evaluate them based on the incidence and severity. Incidence is when we look at the cluster, when we see a single berry is infested, if there are 100 berries on that berry or cluster, we call that the 1% incidence, which is not that big problem as an incidence. But when it comes to severity, which tells us how big the area of the cluster is infested, which is bigger part of it, which is more important than that. So, as you can see, those are the higher. I'm not going to go one by one to all this, but I'm going to tell you that some of them are, as you can see, is a program, which means that start with the sulfur, continue with the synthetic, and then continue with that. So when the disease pressure is high. So we do the industry standard for companies to compare their product with them. So we also have a lot of organics and all kinds of things. This is in 2024. In 2023, we also treated some of the Oro Agri or Rovensa products. They were not 100%, but they were significantly managed the powdery mildew. As a rule of thumb, I'm going to tell you that how soft chemicals work versus the hard chemicals. So I'm going to get into that. That's what I'm trying to point out here. Here's the table of the soft chemicals, which includes biocontrol agents, organic materials, or plant-derived materials. And then, as you can see, incidence and severity is a little bit higher than the synthetic chemical and combinations. As a rule of thumb, soft chemicals work slow but sustainable way. It provides you the long-term management, less of the risk management of the powdery mildew resistance, which I'm going to talk about more on that one. But when it comes to synthetics, they work like a charm, short-term management. It's not sustainable. It has a lot of side issues, we all know, and then that's the difference of that. Now we are trying to give you an idea how soft chemical works, how hard chemical works. If you combine them, they also work together. If you are not organic, that will also give you an idea. So this is the taste of the results that we have been doing it. At the end, I'm going to show you my lab website where you can find those individual reports, including all this information in our website. In this part of my talk, I'm going to talk to you about the bunch rot and gray mold. Bunch rot is caused by the Botrytis cinerea. It is one of the most ubiquitous pathogen that can be found everywhere. Okay, and then their issue is that the difference between botrytis and powdery mildew is that the powdery mildew cannot handle sugar. It attacks the berries or grapevine in early stage. When the Brix level turn into the certain level, above 12, they cannot colonize on the fruit. Because of that, when the berry turn into the certain size, you don't need to worry about the powdery mildew anymore, because even though there is a powdery mildew, they cannot colonize. But from that point on, a botrytis comes in, because that's the one that can consume and like the sugar. Okay, so when they start Brix level turning into the 16 and then higher level, botrytis comes in, which is very interesting balance that. The pathogen is, as I said, very well adapted at all the environmental condition, including the cold condition. Okay, many of the fungi or bacteria cannot colonize and move very fastly and speedily during the colder temperature, except the botrytis. It's also known as a cold storage pathogen. I'm sure you have heard about that, so it can go everywhere. So that's the one of them. Another part of the complex of the bunch rot is that it can also cause summer bunch rot on table grapes, which is part of the reason that production that we have been having in Fresno and Central Valley area, very common from now on. So these are the sour rot, summer bunch rot complex that you can see on table grape, and then it can turn into the slip sicken. Slip sicken is a little bit more advanced part of the sour rot when the yeast and also other acidified bacteria gets into the system, they can start causing these kind of problems. And then this is the one that can get in, and then other pathogen like the Cladosporium also can colonize then, in that area. So those are the some of the pathogen that can get in. When we look at the infection time and period of the botrytis, when a single the cell or the conidia or spores land onto the fruit, it starts germination tube, like similar to the powdery mildew, Botryosphaeria, and then it produces the kind of like the punching point, Haustorium gets into the plant and then start colonizing it. The difference is that botrytis can produce cellulase, which is an enzyme to melt the cell wall of the tissue and then get turned into the melting like that, as you can see. And then within a short time it produces conidia, hundreds of the conidia. That takes 24 hours to 48 hours when the temperature is very good. So remember, for powdery mildew it was almost 1 week. For this guy, it's 24 hours to 48 hours. I don't know if you remember that a couple years ago we had an unusual rain in August in Central Valley. After that rain, growers may not be able to couldn't get into the vineyard to spray something there to protect it, so they couldn't get in for almost 10 days. Because of that, this pathogen can multiply within the 24 hours, so they lost a lot of their crops in that one. As a grower, important part of it is that when you know with the climatic data that's gonna rain, that unusual rain gonna happen, you can apply some pesticide that has a local systemic effect of it, so that may help you the prevention of that kind of after rain, because you are not going to be able to get into the vineyard after for a while. That kind of situation. So it's very, very important for the disease management what to expect, how to act before that expectation comes, which is the preventative. As a rule of thumb again, it is easier to prevent the disease than to manage or cure, because there is not much curative the management system that the sustainable way of controlling is that's the way. My lab has been working on this sour rot complex on not only wine grapes but also table grapes as well. We have been finding not only botrytis, as I mentioned, we have found recently 3 Aspergillus species are involved with this sour rot complex. Cladosporum, Penicillium, Rhizopus, yeast, and bacteria is involved, all of those. So why is it important? If I compare with this disease with the powdery mildew, powdery mildew has a single pathogen. We know what it works, and then apply that. But when it gets into the more than one pathogen complex, each pathogen has different mode of action, different aggressiveness, different lifestyle. That's where the things get messy. You cannot control all of them with single product. You have to be concerned about the multiple, more sustainable, more organic and biologically acceptable way of managing it. That's the important part of the system. Recently, we also identify another diseases that also found out in tests. This kind of symptom in early fall. I'm sure if you are associated with the vineyard, you have seen this kind of symptom that is associated mostly with the viruses. But this particular species has been tested by us and by other laboratories. It was virus-free. No leaf roll tree, no red blotch, none of that. When we look at the trunk of the surface of that infected symptomatic plants, we have found this kind of black structures are just oozing out on the trunk under the bark. This is the close-up picture of it. When you peel the bark exposed to that phloem and then cambium tissue, you can see that kind of like the brownish, some cases purple color of the colonization under the bark area. When you look at the cross-section of those samples, you can see the canker, which is the dead tissue in this area, as well as some tissues here in the active xylem, woody tissues. As a plant pathologist, this is telling us that whatever this causal agent, which I'm going to tell you now, cause canker, and also it could be a kind of systemic pathogen, xylem moving pathogen within the xylem tissues. That's what it's telling us. Why is it important? If it is canker, it cuts and then kills this part of the plant and get rid of the cycle of the plant or interfere the cycle of the plants. But when it gets the xylem and then moving the xylem, there is a chance that it can move from trunk to the other branches and then sometime into the cluster as well. So that's like the dangerous part of it. Temperature, humidity, relative humidity, or sometimes precipitation, rain, water, whatever you name it, all those environmental conditions. As the technology developed, we can only manage environmental condition. Well, we can also manage the pathogen to increase their resistance. That's the kind of resistant part of the things that we have been, the host, pardon, that we can change the host plants, increase their resistance or suitability. Besides the playing with the environmental condition, which could be the leaf removal, is the essential. Dr. Gubler has done a number of the studies that he found out that by just leaf removal around the cluster, by increasing the airflow, that reduced the right environmental condition for the disease in the cluster area, you can save or manage the disease almost 50%. But nowadays, we have to be careful in California conditions. When you expose the fruit to the sunlight too much, you can have the sunburn. So there's a balance, consequences that you need to find the balance on that. Decreasing of the fertilizer. Why is it important? Fertilizer, especially nitrogen, can promote the growth too fast. When the plant grows too fast, it produces a weaker cell wall. And that cell wall is one of the defense mechanism or barrier for the pathogen to get in. So that could be the interest. Instead of decreasing, I will say balance it, because you have to use the fertilizer sometime. Cluster architecture is very important. Some cultivars are more susceptible. Why? Because cluster is so thick and compact that you cannot get any air and condition inside of the cluster. That could also be very advantageous for the pathogen to colonize in that area. Sometimes clusters could be burst because so compact. As soon as a burst comes, that means the available food is out for the pathogen. They can just colonize that and then get into their system in that area. Again, trellis system is the similar idea, reducing the right environmental condition, and then another last one is the fungicide or management strategies. But application of the fungicide or biological or any product is depending on the timing for the botrytis. For powdery mildew, I mentioned you that you have to start applying, looking at the climate data, start applying from the beginning of the season, controlling until the fruit level become not susceptible for the pathogen. But for the botrytis or others, start with the bloom, pre-close, veraison, and pre-harvest. Why is it the bloom time? Because botrytis spores are so small and available everywhere, during the bloom time, it can land on the fruit, flower, stigma, and then act like a pollen, germinate, get into the pollen tube, and then stay there, wait. What is it waiting for? We call that one the dormant or latent season. It's waiting for the sugar level in the fruit to be higher so they can colonize and then consume. So that's the most dangerous part. If you cannot prevent that happen during the bloom time, you are not going to be able to control it because it's already in the pollen tube, and you are not going to have the systemic pesticide easily to get control of it. So you have to prevent it from that. But the percentage of bloom time infection is not that high, but there's a certain percentage that can happen. And the others are very, very important as well. Besides the application time, the coverage is very important too. As a rule of thumb, you may have a silver bullet for a pathogen to eliminate, but if you don't have the good coverage, it is not going to work. You are going to waste your product, you are going to waste your money, you are wasting everything. So earlier talk that mentioned about the coverage is very, very important in the application of the things. So both sides of the wine. So phenology is very, very important. to know what phenology is the more susceptible for each diseases, so that will give you an idea about that. We also set up experiment with botrytis management in the field as well. And so the way that we set up the experiment, we applied the randomized design with the fiber application. We use application in the bloom, pre-close, veraison, pre-harvest, and then we use the steel backpack sprayer, which is the air blast sprayer, which is the closest that we can get with the air blast that the growers have been applying in the art. This is the 2024 powdery mildew botrytis bunch rot results from the part of it. I just wanted to include a couple of the product from Rovensa to have an idea what I have been working on it. And then now it says the typhoon area incidence and severity, and then the control is there about the 40%. When you compare those incidence and severity, all these products seems to be significantly reducing the infection of the botrytis in the plant. So you can also see the other synthetic pesticide with the hydrogen peroxide, Mavalon, which is another the plant-based extract, but it's not organic. So they also shows some efficacy against the botrytis. If you would like to see the full spectrum of this result, again, this could be found in my lab website. Here's another way of showing those results. And then these are the showing that comparing with the control, the Rovensa product, especially the typhoon with the attitude. I don't know if it is the right one of it. It seems to be the effective, not only by the severity, but also incidence of the botrytis as well. In this slide, I'm going to just go through the importance of using biological or organic fungicides. I briefly mentioned about what is the differences between them. Residue management. Nowadays, in Europe, when you go to Europe in the wine industry, they almost abandon all synthetic chemicals. And if you want to register a new synthetic chemical, it's going to take multiple years and up to $1 million money to be able to register, which is not possible. California is almost following that system, getting into that one, and then moving towards sustainable way of management and organic and biocontrol and everything. I'm sure you have been feeling and seeing that kind of what's going ever. Why is that? We all know that the synthetic chemical have a lot of residue. It could be identified in the fruit, and if you don't apply or go with the label rate, you may have the consequences. You have to apply those. But there's still risk of residue of those chemicals on the fruit. Resistance is the most important part of this picture. For botrytis, it's very quick pathogen to produce resistance to isolates, which I'm going to talk to you about that. For powdery mildew, same. Why? Any living organism on this earth has a survival instinct, including fungi. When you corner them, when you push them, when you stress them so much, they will find a way to survive. What are they going to do? They are either going to change the environment of where they are, they cannot change it, they are going to change their genetic structure. If a synthetic pesticide targets one single part of the pathogen, which called single mode of action, that is identified by the FRAC group. In each label that you look at it, they say the FRAC group 1, 2, 3, 4, 5, right? Whatever it is. That tells you mode of action of that pesticide. If that pesticide targets one part of the pathogen is going to be easily develop a resistance because one shot. If I have to give you an example, that think about the two boxer matching, okay? You know that your opponent, boxer, it has a very good right shoot, right? When it is, you know you are going to be prepared, like the right punch. Get ready. Anytime that guy has a very good right punch. But suddenly, if that guy put you the left punch, which is not expected, it's going to knock you down. Single mode of action of the pathogen, the fungicide, is the one single punch versus multiple mode of action, right, left, up, down kind of things. So that will increase your chance of reducing the chance of resistance on the pathogen. That's the importance of the frag group in that one. When you look at the biological frag group, that's a little bit broader than the synthetic biological. For example, strobilurin group of the pathogens, so fungicide, they are not working anymore on powdery mildew because it's a great mode of action, very broad spectrum, better. It's originally found in the nature, and then, but because of the single mode of action, it's not working anymore in certain isolates in that area. So that's a very important how to resist the management of the resistance against that. These are the frag group that I'm talking about. So because of that, when you are looking at the pesticide, pay attention what kind of frag group, and then get more information about what that frag group means, what kind of mode of action they have, single, multiple, or organic. That's the important part of the mode of actions. Sustainability and soil health, I'm not even going to get in, so you get the idea. And then all these marketing is very important. Nowadays, consumers are more aware what the organic is, what the biocontrol is, what kind of product that you look, everything. Everything is in our phone. They just look at it and then get the information and then share those information, which is very, very important, right? To give you a taste of what that the fungi of the botrytis do during the growing season, my lab has been screening the diverse botrytis during the different stage of the growing season on grapevine in California. This is the study that we have done on the table grapes, and then we collected some samples in Fresno area as well as all the areas that I'm going to show you. So what we did with this experiment is that we collected samples of the flower from the different location on the vineyard. So these are the locations. We are right here, and then we collected samples from all the table grape growing areas in that area. When we look into those flowers, what kind of fungi they have, this is what we found. For the flowers, we found Alternaria, most of them, Aspergillus, but mostly Botrytis and Cladosporium. No Penicillium, no Rhizopus. They are the one all come later on. These two pathogens get into the berries or stigma during the flowering time. These are all the locations that we collected. When it turns into the berries, similar one, it turns into the different stem. Aspergillus, that turns into the Botrytis again, Cladosporium, Penicillium start coming up, Rhizophus, and yeast is coming later on in that berries. Okay, So that's showing us the importance of that, when to apply, how to apply those pesticide or fungicide or biocontrol agents in that area. We are also now screening those different isolate of the botrytis to see diversity of those botrytis, the resistance. We found out that number of the fungicide are not working anymore, not effective for those fungicides, so for those botrytis. That's like the interesting part of it that comes out. So far, I give you a little bit more information about the powdery mildew, botrytis. Those are the two most common fungal diseases of the grapevine that you have to be on top of it. Apply the whatever you want to apply on time, preventative matter, and either choose your right product if you want to go sustainable, biocontrol or organic, if you want to go the synthetic, or if you want to combine both of them. There are a lot of studies, if you are not organic, to increase the effectiveness of those biological that works slow and sustainable, people alternate with the synthetic chemicals to increase their sustainability and effectiveness. That's also another way of applying this, to increase the effectiveness of the sustainability in your vineyard. So those are the alternative types. So it is not like old days, whatever you hear from your neighbor, oh, what are you applying? It works? Okay, I'm going to apply to it. That is gone. Why? A lot of companies are working and then bringing up the new products. I'm not telling you to go to companies and then, yeah, okay, whatever you have it, I'm good. No. Do your own research. Nowadays we have a lot of like the smartphone in our hands, and then do your own homework. Hear it, believe it, but confirm it. Investigate on that. So these are the basic informations for you to what to look for, how to make decision. Okay? Because of that, it's important to know what's going on out there, what information is new, and then this is one of the information that we are providing you to do. What are the latest products that could be alternative for you to use? We are not telling you use A, B, C, D. That's not our job. Our job is to give you whatever it is outside, and then provide you the results, and then make your own decision to use in that area. So those are the products that we are talking about that. Do I have time? Should I get into the tripe and trunk diseases? Are we doing good? Is it boring? No? Hopefully not. Okay, so this is going to be like a little bit more different part of it that can. How many of you have heard about the grapevine trunk diseases? 1, 2, that's good, 3, but we are going to go into this. Grapevine trunk diseases that used to be known as young vine decline, ESCA, Eutypa dieback, bot canker or Botryosphaeria canker, Pomopsis dieback, and blackfoot. Now they are all known as the grapevine trunk diseases because they colonize woody part of the plant. Comparing with the powdery mildew, leaf and fruit, botrytis, fruit. Okay? Sometimes botrytis get into the woody tissue, but these are the true wood colonizing pathogens. Some of them could be the vascular. Earlier I told you some of the Aspergillus species could be the vascular pathogen that can move up and down, and then young vine decline and ESCA disease are the vascular pathogen. What does it mean? They can get into this vascular tissue, move up and down, colonize like that, and then when the plant get infected with these pathogens, they respond. They respond with gumming and tylosis, which is this kind of goo coming out. So this disease used to be known as a black goo. Now we call it ESCA. is a symptom that you can see Eska. Very classic. Okay, it's also known as the tiger stripe leaves. And also, this kind of fruit, which you can see in the white varieties very easily, it also occurs in the red varieties, but they are not visible with those specks, measles. When you see those symptoms, you may think that pathogen somehow get into the fruit, colonize there, and then when you apply something on the fruit, you may protect it. No, does not work like that. These are byproduct of this colonization of the xylem tissues, wood tissue, which means that when the pathogen colonizes in the wood, they produce secondary metabolites like toxin. That toxin causes these symptoms. If you want to manage these, you have to get into either prevent and manage those xylem vessels. It's very important to understand biology and life cycle of the pathogen that you are dealing with. Otherwise, you may go and then spray something that may not even work on that. And pesticides are expensive. Eutypa dieback and Botryosphaeria canker, these are the canker pathogens. What does it mean? When they colonize in the wood, they cause that kind of dead arm. It can turn into this, and then these are the cankers, which means that this is the dead part of the plant, this is the living part of the plant that you can see. There is no reverse when the contamination happens. Pomops is similar. It starts with the green tissue, especially this year we have been seeing more because we have more humid and rainy days. And then it can also turn into the canker. And then black foot is another complex, which is soil-borne pathogen. It used to be known as a cylindrocarpon. Now it's known as the Ilionecria, and then that also causes that area. What are the differences between powdery mildew botrytis versus the canker pathogen? For example, for powdery mildew, if you cannot control powdery mildew this year, you are going to lose your fruit. You are going to learn your lesson, and next year you are more proactive. You can control and then be a good production, no problem. Just lose the product at that year. But when it comes to grapevine trunk diseases, when they get into the plant, it is very, very difficult to stop and control them. At the beginning of the stage, management, preventative management is more important for this disease than any other diseases. That's if I have to compare you to those important. If you cannot manage it within the 5-10 years, you are going to have the good product, no problem. But after 10 years, within the 10 to 5-15, you are going to have the 75% increase of these diseases, which is the most productive time of the grapevine vineyard. That's how dangerous they are. These group of the pathogens that were adapted environment so good and better, they produce kind of like the fruiting body on the trunk, which is very easy, that can spread from one trunk to another trunk by air, which I'm going to talk about that. These are the close-up pictures, and where the canker is, that you can see all the black spots. That's the kind of their seed source of the pathogen. These are how it looks like in the close-up. When you put a single drop of water and a precipitation, that can activate that structure, and then each structure produce or release thousands of the spores, which each one of them is like a spore or seed of the pathogen. This is the asexual fruiting body. The plant or pathogen produce each one of them identical and seriously. Same pathogen could also produce a perithecia, which is the sexual reproduction of the pathogen, which I'm going to show you a video. For Pycnidia and perithecia, if you have to compare, Pycnidia produce release, passive the spread. Paresthesia actively shoot the spores in the air that could move very fast. Isn't it a very smart way to survive in this world? They have been adapted and managed how to produce, how to get the less chance to not to survive, how to get the higher chance to survive in this world. So that's very, very important. So why is it important, this information? If you know how your pathogens release, spread, when to spread, how to infect, all this information is going to help you to make decision how to prevent them. So having knowledge, information about the pathogen disease is more important than knowing a fungicide and then grabbing and then applying it, because otherwise it's not going to be sustainable way of it. We have done a lot of spore trap studies to find out when and under what condition those spores release in the air. When we look at this is one of the example, from during the season or year, January to December, not season, year, these blue lines are showing the precipitation. I say precipitation, not rain specifically, because at dew point, fog could also activate these fruiting bodies. And then look, whenever we had the precipitation and rain, we trapped spores, which is the bar in this area. From May to October in here, Sonoma, there was no rain, no spores, and late October, December, more rain, more spores. It's very, very correlated with the spore. What does that mean to us? Why this is very important for us? So what we do during the winter is that we prune our vineyard or grapevine for the dormant pruning. We make a significant amount of the wound. Any wound could be open entry point for those pathogens to get in. But we cannot go ahead and then prune during the summer. That's not possible. That means that we have to do something during the winter time when the spores of those pathogens are in the air available so badly. Another question is that how long those pruning wounds are going to be susceptible after my pruning. When I prune it, how long that I can be safe after the pruning. These two graphs are showing that, and there are many studies that has been done. Pruning wounds could be susceptible more than 1 month. So that also doesn't give you an idea, window, hey, let me apply this one during the dry season if it's not raining, and then be safe. It doesn't work that way. So you have to do something actively to prevent those pruning wounds. How do they infect the plant? Primary entry point of these pathogens are pruning wounds during the winter, period. Almost 85% of it. If you can protect those pruning wounds, you could be most likely be safe in that area. What other way of going that? They could be also latent and endophyte. What does it mean? Some of these pathogens, very interesting, that could be present in the healthy tissue of the plant, but not causing any disease. They just stay there. Plant defense mechanism is good, life is good, everybody is happy, no problem, that's okay. In a summary, from all these years experiment that we have been doing, now we have alternative biocontrol agents, including number of the Trichoderma species, one beneficial Arabasidium species, and Bacillus velazensis, which is the beneficial bacteria, are working to prevent the pruning wound. They are more sustainable way, which I'm going to show you why they are sustainable way. We also try a plant extract based on the thyme oil. It also works and then prevent that one. Number of the synthetic fungicide, if you want to go with the synthetic. There are sealants and also disinfectants that are working. Again, I just want to make sure that what is the difference between biological and then synthetics in this graph. This is the graph that shows the mode of action of each individual beneficial. For trichoderma, beneficial fungi that could be actually present anywhere with the right condition, mostly or primarily work as a competition, which means that those beneficial fungi can colonize on the surface of the plant so fast, when the pathogen comes, they cannot find a place to colonize. Competition. They also have endophyte, antibiosis, and go on. Keep in your mind, that's one of them. It looks like that they have 5 different modes of action, which means that the work of these beneficials are more than one punch. When it comes to bacillus or bacterial-based beneficial, in this case the bacillus beneficial, they also has the multiple mode of action, but the majority or primary way of preventing is that the antibiosis, which means that when they colonize on the tissue, they produce a secondary metabolites, which is the toxin for the fungi that they cannot colonize. Here's the downside of it. You cannot apply both of them at the same time. Why? Because this is a fungi, it doesn't matter beneficial or harmful. This is going to produce antibiotics to prevent that. So you got to be careful what we are talking about. Sometimes we just get greedy. Okay, I want to apply both of them at the same time to get the benefit. No, it doesn't work that way. Here's another benefit of using the fungal-based beneficials. When you apply them 1 year and multiple years, you are naturally increasing the beneficials in that vineyard. How do we know? In our lab, one of my graduate students did this study. We collected naturally occurring trichoderma species in the California vineyards, identify a bunch of other trichoderma species, which here is known as the UCD. They are also working very well comparison. So that means that if you can increase those beneficials in your vineyard, in a long time, you are going to use less pruning wound protection. We are going to have the natural occurring those beneficials in a long time. So that's like the beneficial part. We also found out that we were able to re-isolate those beneficial trichoderma from the pruning wounds after 4-5 months application. One of my colleagues from South Africa has done the similar studies. They were able to recover their beneficial after 8 months. So depending on the climate condition, those beneficials could stay on that pruning wound longer time. Everything that I told you, results, reports, that can be found in my lab website under this link, Fruit Crop Fungicide Efficacy Trials. Everything is there, and then it's for you to download, read, whatever it is. I wouldn't recommend you to read those scientific papers that also you can find there. Because, well, you can read it, but you should start reading before you go to bed 10 minutes. It's going to make you a good sleep. With that, so scientific information is going to be that kind of things. But we have a lot of good pictured and information that's not going to cause the same effect of it. And how are you going to find that? These are the results that you can find, each one of them. We are not only working the grapevine, but also other fruit crop fungicides trial. And then every year, this year is going to be, every year we have the field day to show you those results on our campus, on the vineyard. This year, the field day is going to be July 18th. You all are invited. You are welcome to come, join. It's in the morning. You can find that information in my lab website as well. It's free of charge. We also provide a free lunch if you like, and then that's the way. So if you cannot make that field day, when you go to our field fungicide trial results, click on those pictures, you can also see each picture of the dose, each treatment, how they respond by the picture. So that will also give you a visual result of them. With that, so this is my lab website. These are my lab members. These are the funding agencies. And then all this information that I share with you. So thank you very much.